Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Lara, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Lara Nascimento-Brooks from Sartorius Stedim Biotech.
Thank you, Leah, for the introduction. I'd like to thank BPI for the opportunity to discuss with you how to enable increased performance, robustness, and safety in single-use bioreactors. The three top important manufacturing trends in Upstream focus mainly on increased productivity, which can be interpreted as increasing the efficiency and getting the maximum possible information from each batch. Continuous processing with the intensification of fed batch processes and perfusion optimization mainly. And the third is the single-use systems enabling new sensor technologies to be easily implemented and integrated in the system. My presentation will focus on the single-use consumables, particularly for these large commercial processes. In that sense, choosing the right bioreactor concept for optimal performance is paramount. It's important to address the critical parameters for successful processes, like mixing efficiency, power input per volume, and oxygen transfer rate. If you look at the right-hand side of the slide, you will see there is an optimal design space. If we increase too much the bioreactor agitation, for instance, in other words, the power input in the horizontal axis, we may induce shear stress. However, the design of a bioreactor needs to accommodate a minimal viable agitation in order to avoid heterogeneity risks. Alternatively, if we increase too much aeration, represented in the vertical axis in liter per minute, we may induce foam risk. However, the design of bioreactor needs to accommodate a viable aeration to avoid oxygen limitation risks or excess of CO2 in the medium. Therefore, finding the right balance of process parameters in the design space requires high-efficient systems for optimal productivity and a low risk for a safe and reproducible process. In order to tackle these challenges, we must ensure a robust design of single-use bioreactor, and that going from small scales to large commercial settings. Ensuring a geometry similarity from small scale to large scale vessel. For instance, maintaining vessel height per diameter and impeller diameter per vessel diameter can ensure that mixing and oxygen transfer rate coefficients remain consistent throughout the development of a process. Sartorius Amber and Biostat STR platforms, for instance, take that in consideration. Not only does this geometry similarity simplify technology transfers between scales, but it can also mean shorter time to develop a process to reach market. The geometry similarity is not only the only part of the equation. In order to enable high performance, we would also need to look into the configuration of a bioreactor. For instance, by enabling the sparger combined with micro and ring parts can optimize higher oxygen transfer rates while optimizing CO2 removal from the media. The vertical steer shaft impeller, for instance, is a configuration that also plays an important role with specific designs optimized for mammalian, microbial, or microcarrier process. Having a strong characterization of the bioreactor based on the DECEMA guidelines and also computational fluid dynamics, it's of vital importance when choosing which design to go to. A bioreactor should also enable different feeding strategies to be developed, particularly regarding fed batch and perfusion strategies with ports in the lateral side that enable ATS or TFS, smooth connectivity options. Flexibility regarding connectors and tube dimensions is also very important, which is taken seriously by Sartorius with the FlexSave STR bags, easy to define configurations. Having a harvest port at the very end and the very bottom of the vessel, enabling maximum amount of liquids to be harvested is also very important to enable smooth transition towards downstream process. And last but not least, the sensor ports are also of vital importance in bioreactors in order to get the maximum amount of data per batch. Regarding sensors, I would like to focus on the single-use technologies we currently have, because the single-use sensors remove that need to autoclave probes and insert these probes in the reactor bag chamber. They remove significantly the handling efforts and potential contamination risks, increasing thus not only process performance, but also safety. Moreover, by implementing single-use sensors, we can establish feedback loop controls that ensure a process control that is reactive, meaning avoiding deviations and potential batch failures. To give you a few examples of the latest developments, I'd like to show you the Sartorius BioPath foam sensor, a sensor patch based on capacitance that enables the detection of foam inside of the bioreactor bag, and it can easily be positioned at the most suitable height outside the bag. I would like to show also the Sartorius BioPath Spectro, recently launched. It's a single-use sensor based on inline Raman spectroscopy, which enables processes to be monitored regarding their metabolite concentrations and cell densities, and to be used in a feedback loop controls as well. Sartorius FlexSave pH electrochemical probe is another single-use innovation we have in the market with a wide measurement range from pH 3 to pH 10, and with only 1-point calibration, combining the benefits of multi-use traditional autoclavable probes with the convenience of the reduced setup time and effort of the single-use approach. And finally, the BioPath biomass sensor we have developed with our other ongoing partnership, enabling cell densities to be monitored online, which reduces the amount of samples taken by operators and overall handling efforts to monitor cell density. It's a principle based on radio frequency impedance, which enables a measurement of capacitance in the media, which correlates directly with cell density. It has already been well established in the pharmaceutical applications with customers like Bluebird Bio, using the technology to control cell densities in order to increase cell productivity, and also Biogen using the biocapacitance to automate feeding strategies in a better way. But not only sensors are important when considering performance and safety, and also robust processes. For safety, we also need to consider the biosafety considerations that we recently developed an FMEA study upon. In this study, there was an evaluation of different factors that can also impact the safety of a process and also for the operators. Factors such as contact with high temperature, sharp objects, friction, and overpressure are conditions that need to be taken in consideration while handling single-use bioreactors in manufacturing facilities. Without considering these aspects for bioreactor design, these conditions may lead to the rupture of the bag, which leads to leakage and potential contamination in the BSL area, or even projections that could impact operations and operator safety. Therefore, manufacturing companies must be mindful of this risk. In order to mitigate these risks, for large-scale single-use manufacturers, it's important to consider a protective clean room compliant packaging for single-use bioreactor bags, which themselves need to be able to be integrity tested at point of use. For intensified processes, it's also important to protect the exhaust filter line against blockage with the exhaust single-use coolers, for instance, for top intensification performance. And finally, a bag that is easy to install even at large scale as 2,000 liters, facilitate operators' tasks, enabling also an easy handling procedure by the operators. We can also increase safety by searching to invest in assurance of quality supply. In that sense, suppliers are asked to better characterize the raw materials and improve process controls throughout the supply chain. This means from resin and film production, bag component and assembly manufacturing, until the sterilization process finish. From Sartorius' perspective, it's important to have frequent samples regarding bioburden, endotoxins, subvisible and visible particles, as well as ensuring integrity testing at point of use. These practices, of course, lead to a safe and convenient single-use bioprocessing with optimal cell growth, robustness, and consistent extractables and leachable profiles. When choosing a supplier for single-use consumables, quality and performance goes hand in hand with a robust change control and the need for long-term business continuity. In that sense, Sartorius strives to implement a global manufacturing network present in most continents while keeping control of high-standard performance manufacturing throughout the consumable business, from upstream to downstream, future-proofing thus the supply safety needs for the biomanufacturing challenges ahead. This brings me to the end of my presentation, and I would like to thank you very much for your time and attention, and I'll look forward to answer your questions. Thank you.
Great. Thanks, Lara. So the first question is, how do you characterize a bioreactor bag in regards to performance and robustness?
Right. So this is a good question. Performance is evaluated under application conditions, such cell culture, using typical cell lines like CHO cell lines. We also evaluate mixing time that takes to homogenize bioreactor, and KLA studies, which is the oxygen transfer rate coefficient studies. We can also evaluate the robustness under worst case conditions. And in parallel to that, we evaluate accelerated and natural shelf life of the bag, considering the assembling studies and other packaging integrity studies.
Okay. And is the pH electrochemical probe able to be stored dry?
Yes, so the pH electrochemical probe is provided in a dry format. So when it's integrated in the bag, and throughout the life, integrated in the bag until point of use, is protected with a thin layer of colloidal material that protects the overall sensor.
Okay. Is the biomass probe applicable for microbial processes and mammalian processes?
That's a great question. Yes, it is. It is possible to be used in a wide range of processes, including microbiome as well.
And how is Sartorius preparing for the new USP 665 guideline?
That's a very good one as well. The USP 665 is under preparation by the authorities, and its draft version is in review by our quality teams who are working on this. We still have a bit of time, as it will be enforceable by 2025 only. And we are aware of the 665 chapters. They are integrated in our regulatory survey. So we are following the changes and the official publication when it comes.
Okay. And can you speak to the mixing performance of the single-use bioreactor bag, as it's not cylindrical like a stainless steel bioreactor? How does the shape affect this mixing performance?
So when we developed the design of the biostat consumable bag. We based our development on the traditional stainless steel bioreactors. Of course, there are some differences in terms of the bottom assembly, and these differences are linked mainly to the process of assembling the bag and make sure that the bag corresponds to the stringent regulatory aspects related to robustness, as I mentioned. But we do characterize our bags, as I mentioned, through competition fluid dynamic analysis. And thanks to the vertical steering shaft impeller, we see the performance very equivalent to the traditional stainless steel bioreactors, not only for the mammalian processes that we evaluated, but also microbial processes and also microcarrier processes.
Okay.
And then I think the question, sorry, just reviewing the question, it is related to the mixing performance. So mixing performance is one of the things we also evaluated in our performance testing. Not only the oxygen transfer rates, but mixing is also very important. And we do evaluate mixing times. Currently, we have mixing times that are below 20 to 30 seconds throughout the range from 50 liters to 2,000 liters.
Okay. Do you have stability or drift information or validation for single-use probes, Raman pH, and for the BR bag itself, especially for perfusion processes that continue over time?
That's a great question. Yes, we do have studies performing comparison studies between the traditional autoclavable probes, not only for the pH, also Raman, and comparing that with the single use. So we could definitely share those. And our focus really is to go to longer periods of time to cover those processes like perfusion. So we have evaluated also these probes under intensified conditions as well.
Okay, very good. Well, I think we have time for just one question. As a reminder, we will be passing any questions we didn't get to Lara directly, so don't hesitate. Go ahead and you can continue to type in your questions. But I think we just have time for one more question for the webcast. So the last question is, are you doing anything differently with your STR bags for ATF processes?
Yes, so we adapted the lateral ports, accommodating up to 1 inch wide in terms of diameter to enable ATF and TFF easy and smooth connectivity. Okay,
great. Thanks, Lara.
Thank you, Leah.
And thank you for joining us. As a reminder, the recorded version of this webcast will be provided. And as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.